Okay, so it's my great pleasure to introduce Emily Dolson to give our ICSA seminar this week. I've known Emily for several years now through prominent member of the artificial life community. So we've met at several conferences and other events, and she does some very interesting work in stuff that should be of great interest to a lot of ICSA people on artificial life, and today on controlling the course of evolution, which if you can do that, that's really exciting. So over to you, Emily.
Awesome, thank you. Well, we can't do it yet, but hopefully eventually. Yeah, let me just share my slides. Okay, that work okay?
Yes, that's fine.
Awesome. Okay, so hi everyone. Thank you so much for having me. I'm so excited to be here. This seems like exactly the cool interdisciplinary group that I love talking to. So yeah, as Susan said, today I'm going to be talking about some preliminary efforts towards being able to control the trajectory and speed of evolution. So hi, I'm Emily. I'm an assistant professor at Michigan State University. I run the evolutionary control of digital ecologies lab, and I have been for all of like 6 months. So side note, if anyone has undergrads or master's students that are looking for PhD positions, I am definitely hiring if this sounds cool to you. So just a quick overview of what I'm going to talk about. So because it sounds like this format has a bit more discussion at the end than a lot of talks do, I wanted to spend a little more time on the introduction than I usually would, just to give you kind of a lay of the land of the full space of the problem I'm trying to solve. And then I'm going to focus in on 2 specific stories about trying to solve portions of that problem. And so one of them is kind of going to be the neat, elegant, top-down type of solution. And the other one is going to be the messy, bottom-up beginnings of understanding another part of this problem. So those will be kind of a nice contrast with each other. Okay, so let's dive in. Oh, also, I should say, if anyone has questions or anything, feel free to ask them in the chat. I'm pretty used to it from enough online teaching at this point, and I want to make sure everything makes sense to you. And I know this is a very interdisciplinary audience, so tell me if I'm like using some field-specific jargon that you don't know what it is, because I would love to explain. Okay, so the long-term goal of my research as a whole, so my goal for my career and like maybe the careers of some of my grad students is to be able to steer evolving ecological communities to target states. So what do I mean by that? Well, basically, there are a lot of situations where we've got populations of multiple different species that are all part of the same ecological community. And so they're exerting ecological pressure on each other at the same time as all of those populations are evolving by natural selection. And so these ecological and evolutionary dynamics are interacting with each other and working together to produce the end result of this evolutionary process. And so if we want to be able to predict and perhaps ultimately control where this population or community of populations ultimately ends up, we need to kind of understand these underlying ecoevolutionary dynamics and what their interplay is going to be. And why do I care about this problem? Well, it turns out there are a lot of communities in this situation that it would be very useful for us to be able to control. So one of the simplest is evolutionary computation. So in evolutionary computation, we're basically using evolution as a machine learning algorithm. So we've got a population of possible solutions to a problem, and we select the ones that are closest to solving the problem and make copies of them with mutations. And so we keep doing that, and over time, because evolution works, we are able to solve the problem. And so this is a kind of simple example where we're evolving an equation that could maybe relate an input to an output, but this can get much more complicated. And I'm going to explain in a bit how that's an ecology, but so trust me for now that it is. And it would be very useful to be able to guide it to a target state, because we want that target state to be the state where it's solved the problem. And so clearly being able to control evolution in this context is useful. But a completely different context where it would be just as useful is here in cancer. So this is something I worked on in my postdoc. So sometimes we think about tumors as being a collection of copies of kind of the same cell, but really they're not. Most tumors have a lot of different types of cells that are in them that are working together to create an ecological community that supports the tumor. And so these cells are also evolving on a rate that is very relevant to us because they often evolve resistance to drugs and other treatments. And that's why we aren't able to cure cancer very effectively. And so this is another situation where we would very much like to be able to guide this evolving ecological community to a state, in this case, one where it is dead, or at least where we can kill it. Yeah, another example of kind of a micro health situation is the gut microbiome. So, you know, we're increasingly learning that the gut microbiome and other microbiomes within our body are incredibly helpful to human well-being. And we don't totally know yet which states are associated with better outcomes, but we're getting an inclination that some are. And again, this is a situation where the evolution is happening on a timescale that's relevant to us because these are microorganisms living in our bodies. You know, my gut microbiome has probably evolved to me in some sense, to some extent. And so again, being able to kind of influence the trajectory that it's going to take would be really useful. And then lastly, and this is kind of the moonshot, no idea if this is going to be practical, but it is kind of the thing that ultimately drives me and my interest in this work, is kind of macro scale terrestrial ecosystems. So forests, prairies, you know, all the biomes you learn about in school. So as climate change progresses, these ecological communities are going to be becoming increasingly unstable as their members need to adapt to the increased temperature in order to be able to survive. And we depend on a lot of these ecosystems for our own survival. So it's really important to us that they continue to function. And so what I'm ultimately hoping this research can allow us to do is guide these large-scale ecosystems to a state where they can survive in our new climate and they can help us survive. Okay, so that's the high-level motivation for why we would care about this problem. How do we solve it? So this probably seems like it's just a gigantic, overwhelming problem that, like, how is this even like a single research focus? It's too big. So now I'm going to break it down into a set of steps that theoretically we could use to solve it. And this is going to be a vast oversimplification, as I'm going to elaborate on in a second. But just to kind of give you some hope that this could happen, this is what a solution might look like. So the first step in this very simple way of solving this problem is to find a set of fitness landscapes to swap among. And so what do I mean by fitness landscape? Well, this is a metaphor that we use in evolutionary thinking a lot, just in case any of you are unfamiliar with it. So basically, this is a very simple fitness landscape that I'm showing right now. It's got two traits on the X and the Y axis. So these are just labeled as combinations of genes, but you could think of them as any kind of trait that an individual could have, like height and eye color or something else. And then the Z-axis here is the fitness of having that combination of traits. So it's probably not height and eye color because I don't really expect those to have like an interaction like this in the fitness that they give you. But you know, there's some traits that interact in some way. And this metaphor is useful because we kind of have this intuition that we expect populations to climb hills in this fitness space. Because as you get higher, you're fitter, which by definition means you're more evolutionarily successful. And so the population is going to go in that direction. So like I said, this is a very simple one. In reality, any fitness landscape that actually exists in biology is going to be thousands or more dimensions. So they're very challenging to actually visualize. So it's not a perfect metaphor, but it's one that is helpful for thinking about this type of problem. Okay, so that's my detour explaining what a fitness landscape is. Again, if that didn't make sense, please let me know in the chat. So I said that the first step here would be to find multiple fitness landscapes to swap among. So what do I mean by that exactly? Well, basically, when we think about kind of subjecting an evolving population to different pressures, we can usually think about those different pressures as fitness landscapes. So this is an example of 3 different antibiotics and the E. coli fitness landscape that they produce. And so this is a slightly more complicated fitness landscape than the one on the previous page. So that one had 2 traits. This one has 4. So these are 4 genes that are associated with antibiotic resistance in E. coli. And the genotypes shown here are all possible combinations of those genes being present and absent. So up at the top here, the 0000, these don't have any of those genes. And then at the bottom, the 1111, those have all of the resistant genes. And then in the middle, we've got combinations. The arrows show which genotypes are adjacent to each other. And the color shows the fitness of having each of these combinations of genes. And so you can see that E. coli in these different drugs are on different fitness landscapes. So if you had a population that had none of these resistance alleles, so it was at the 0000 genotype, and you applied this drug, you'd kind of be expecting to nudge it down, in this direction. Whereas if you applied this other drug, you'd kind of be expecting to nudge it maybe over here. And so kind of start to see how we could apply these different fitness landscapes as a pressure to move the population through this genotype space, which is our ultimate goal. Okay, so that's the first step. We could find these fitness landscapes. And then once we've done that, we kind of need to be able to know what is the effect of switching between them. And so this will be a combination probably of a lot of wet lab experiments and also more theoretical computational work. And so when I talk about the effect of switching between landscapes, it's important to realize that this is an effect for like each pair of landscapes or sequence of landscapes. So just as an example, this is some super impressive and labor-intensive work done by Jeff Maltus, where he took E. coli and he started evolving them in a bunch of different drugs. And then for each of those drugs, he tried switching and evolving them in a different one of the drugs. So basically there was kind of a pre-evolution step and then a testing evolution step. And at the end, he looked at the effect of that sequence of environments on the resistance to the first drug. So he was looking at resistance because that is a variable that we care a lot about in studying antibiotics and bacteria, because antibiotic resistance is a huge problem. But we could just as well be measuring any other trait that you might care about evolving. So it could be resistance to heat. It could be yield of corn. It could be anything that you're interested in guiding the evolution of. The key is that we want to know what the kind of sequential effects of these different fitness landscapes are, because starting in one fitness landscape and going to a different one is not the same as the effect of having done that with a different landscape in between. So we need to understand this data before we could think about trying to do this whole process. But so assuming we can get all that data for the problems that we're interested in being able to steer, the next step would be to then figure out how we can sequence those landscapes in order to finally guide our population to the target state. And so theoretically, if we've got a set of landscapes that we can use to push the population around, and we've got the right set of fitness landscapes, it should be possible to identify the sequence. And there's been some preliminary work on that using these E. coli antibiotic resistance landscapes. So this was all theoretical, but it was using the data from the fitness landscapes measured empirically from the E. coli. And basically they were able to show that, yeah, like by determining which sequence you apply these antibiotics in, you can be relatively confident of which state you're going to guide your bacteria to. So here they were starting with a population that was spread across all of the genotypes. But by applying SAM and then ampicillin, they can be pretty confident that they'll get it down to this 1111 genotype. Whereas if they'd put ciprofloxacin in the middle instead, then they would have been pretty confident they'd end up here instead. So this suggests that this kind of like sequencing of environments as a way to steer evolution is possible. So that's really encouraging. But there's a slight problem, which is that that work all kind of assumes that we have infinite time. It doesn't tell us how long we need to leave the population in each of those environments in order to get it where we want it to go. And so that is kind of the last ingredient here in being able to, in a very simplistic way, write a prescription for how to steer a population from a starting state to a target state. Theoretically, if we could do these 4 things, we might be able to do that. Of course, each of these 4 things is incredibly challenging to do in and of itself. And that is even before we think about the additional complications to trying to take this approach. But it is possible. Again, definitely, if any of this isn't making sense, ask questions, because I can't see your faces, so I don't know if you look really confused. So like I said, that was a simplification. There are some additional complications to using that approach to solve this problem. So the first is, I said, oh, we just swap the fitness landscapes in and out. But in reality, that's not a thing that you can just do. It's a lot harder than that. And there's a few reasons for that. One big one is spatial heterogeneity. So if you think about a population anywhere in the world, it's probably not actually just on one fitness landscape. So, you know, if we look at this picture, for instance, a tree that's living here in the middle of this sandy patch is on a different fitness landscape than a tree living down here at the bottom of the slope where the soil is different. And so when we try to talk about the fitness landscape that something's evolving on, we're really talking about some weird average of the entire set of fitness landscapes the population is evolving on, which, you know, are probably like connected in some weird spatial way, and it gets very messy very fast. So spatial heterogeneity makes this complicated, although it's potentially a useful tool. And then similarly, temporal heterogeneity has all the same issues as spatial heterogeneity in that, you know, the fitness landscape a population is being subjected to is also changing over time. But it's even worse than that, because we don't have necessarily as good temporal control of what fitness landscape we're putting a population in as we think we do. So this is an image from some work by Ishan King, who's an MD PhD student at Cleveland Clinic that I'm helping to supervise. And he's been working on this problem that basically all of our models right now of evolutionary medicine are wrong, because all of them operate on the assumption that we can take one drug, give it to a patient for a period of time, and then immediately swap in a different drug as, like, you know, a binary on-off switch. But that's not how giving people drugs works. When you take a drug, your body takes some time to metabolize it. There's this whole pharmacokinetic curve is the technical term for it. And so you're not just going from no drug to an exact amount of drug. And so that has an effect on the efficacy of these protocols. So there are all kinds of practical complications in trying to do any of this. Another complication that is kind of a trade-off, it's got some good things and some bad things, is that when we talk about manipulating the fitness landscape, sometimes we're not just changing the fitness landscape, we're also manipulating other variables too. So this is a picture from some work I was doing during my postdoc on evolving cancer at different oxygen levels. And so swapping out the oxygen level is definitely swapping out the fitness landscape, but it also has other important population dynamic effects. So in this case, lower oxygen reduces the density of the population. And as a result, via some interactions with the way cells decide when to divide, we actually end up with just faster evolution overall in lower oxygen areas because the cells are dividing faster because there's more space. And so that just completely changes the dynamics of evolution in that region. So you have to be really aware of when you're changing parameters other than your fitness landscape. In a lot of ways, it's good because it means you have kind of more knobs to turn in order to try and control what your population is going to do. But it is more stuff to factor in as well. So that adds more complexity to trying to solve this problem. And then lastly, and this is what I'm going to talk about in my second story of the day, is the fact that so far I've been talking about exogenously imposed fitness landscapes, where we like say, okay, I am taking this fitness landscape and I am putting this population on it. But in reality, a lot of fitness landscapes have feedback loops where the population affects what the fitness landscape looks like. And so this happens basically anytime that you have ecological interactions, because ecology is by definition interactions between members of a population. And so, this species is going to be fitter because this other species is present. And that means that basically where the population is on the fitness landscape is changing what the fitness landscape looks like. And so that obviously adds all kinds of messy feedback loops. And as a result, makes the system incredibly complex and hard to predict. So that is going to be the second thing I'm going to talk about. And then the last complication is just an important caveat to mention, which is that in a lot of cases, we can't possibly know the entire fitness landscape. And there's some work ongoing to try to deal with that problem and be able to figure out some stuff about how to steer the trajectory of evolution anyway. But the whole idea of, oh yeah, we just find the fitness landscape is in many ways unrealistic. Okay, so those are the caveats. If there aren't any questions about anything that I just said in the introduction, I'm going to go ahead and move on. Oh, question?
How you doing? Sorry, I was late.
Oh, no worries.
No, I missed the beginning, so apologies for that. But you were just talking about things happening as you change over the slide before this one. I keep going back forward now. Oh dear, there's a bit of lag. You were talking about changing the slide. Next
changing
slide, I think it is. Sorry, I'm really screwing things up on it. There, that one.
This one, okay.
So what would happen if, when you did this changeover, if you changed it over back to the same state you were in before you did the changeover? So you sort of did a neutral change to see the effect of those changes. Have you done that, and did you observe anything?
Oh yeah, that's a good question. So let's see, have I done that? So I think probably most situations where I've been kind of trying switching between fitness landscapes, I've had at least the control where I was switching just to the same fitness landscape. Yeah. Or wait, did you mean switching from A to B and then back to A, or
No, it was what you were saying first. It's the act of the switch. I was just wondering if you could control for that and see the effects of that switch. So going through a switch process, but moving from landscape A back to landscape A in one step.
Yeah.
All of the activities that you would do to make that change.
Yeah, that's a good question. So yeah, I think most experiments where we've tried this, have had a control where we've switched to the same environment, because you're right that totally changes the population dynamics. Yeah. So far, that hasn't made a huge difference, but you're right that it's really important to think about.
Yeah. Okay. So you can then say that the act of switching is negligible compared with this other stuff, or you can. Yeah, it backs up that idea anyway, at least.
Yeah, definitely.
Great. Okay, thank you.
Awesome. Yeah, thanks. Yeah, are there any more questions? Okay. If not, so yeah, first kind of mini story I'm going to talk about is, like I said, going to be a nice, maybe straightforward isn't quite the right word, but elegant top-down situation where we're taking a relatively simple system and we're exploiting the fact that we have good knowledge of it to get even better control, which is very much going to be the opposite of the next story I'm going to tell. And so this is going to be about controlling the speed of evolution. So this is dealing with that fourth step in that list that I gave you, which, just to situate ourselves, I was saying, we can indeed steer evolution by applying these fitness landscapes in sequence, but there's a problem, which is that this only works as time goes to infinity. And we do not, in fact, have infinite time when we're trying to give patients drugs or stop climate change or evolve evolutionary computation solutions, any of these things. And so it would be really good if we could make this not take infinite time, and also just know how long it's going to take. Because right now the problem is we don't. And fundamentally, the reason for this issue, like why it arose mathematically, is that basically this math was all done by assuming that we had a population that was at the equilibrium distribution for a given drug. So what I mean by that is for every fitness landscape, there's going to be kind of an equilibrium proportion of the population that will have each genotype. And that's where you would expect the population to eventually settle if you left it in that fitness landscape for infinite time. And so the question is really like, how long is it going to take the population to reach that equilibrium state? And can we make it reach the equilibrium state faster? Okay, so can we do that? Can we speed it up, eliminate this lag between where the population is when we immediately have just changed the fitness landscape, and put it into its equilibrium for that fitness landscape quickly so that we can move on to the next fitness landscape and the next fitness landscape and so on. So spoilers, it turns out we can. That's why I'm talking about this. And so the things that we need to know in order to do this are kind of our standard evolutionary parameters. So the mutation rate, the population size, and so on. We need to have good knowledge of the fitness landscapes of the changing environments that we're going to subject the population to. And we need to already have that infinite time solution, because basically what this advance is going to do is it's going to eliminate the lag that occurs at the point where you change the environment between the distribution of the genotypes and the equilibrium for that new environment. It's not going to magically steer evolution using a set of environments that couldn't otherwise steer it. Like, this is solving the time problem, not the steering problem. But if you have those things, the solution will work. Okay, and so what is the solution? It's something called counterdiabatic driving. It's going to be a little bit mind-bendy, so I'm going to talk through it slowly, but it's really cool that it works. So counterdiabatic driving is actually this idea that was developed a few years ago in quantum physics. So this paper was a super interdisciplinary effort. It was a mix of mathematicians, physicists, me as a computer scientist, and some cancer biologists, all coming together to try to solve this problem. It was a really fun group to work with. So the basic idea behind counterdiabatic driving is that it is a way to take a noisy system that is in one position and move it to a different system. And so these were originally quantum systems, because those are certainly noisy systems that you would often move between different points. But an evolving population is a system just like this. Because of all the mutations, there's constant noise. And these different distributions of genotypes are different positions that you might want to move it between. And so normally when you think about moving a noisy system, there's trade-offs, because the faster you move it, basically, the more noise you're going to introduce. If we think about this issue of applying drugs in sequence, or any fitness landscapes in sequence, like I said, if we apply the next drug before we've reached the equilibrium for the previous one, we're going to add further noise to the system, because we're going to have a part of the population that wasn't at the genotype that we thought it was, and who knows what path it's going to take through the fitness landscape. Whereas we'd like to be able to move through these different fitness landscapes quickly without introducing that noise. And that's what counterdiabatic driving is going to let us do. So I'm not going to talk about the math behind counterdiabatic driving, because it is intense quantum physics math, and I cannot claim to 100% understand it. That was the job of Mike Konchesky, the physicist on this paper, and also Shamreen Aram, who's a PhD student in his lab, and handled a lot of the math for this. But what I am going to talk about is the intuition behind why this works. So let's for a moment think about a waiter carrying a glass of water on a tray. And the waiter presumably wants to carry the glass of water to a table. assuming one day in the future we'll have waiters and tables in restaurants again. And so if the waiter wants to carry the water without spilling any, one solution would be to hold the tray perfectly flat and walk infinitely slowly. This is obviously not ideal because, again, we don't actually have infinite time, but it would work. If the waiter tried to move faster than infinitely slowly, then the air resistance and other forces would act on the glass and it would start to slosh around and potentially spill. So that would be a problem. That would be exactly an example of noise getting introduced to the system when we try to move it faster. So what do waiters do in reality? Well, they tilt the tray as they're carrying the glass. Some do it incredibly impressively. I feel like I've seen them like hold it completely vertically. And basically what that does is they're holding it in a way that perfectly compensates for the other forces that are acting on the glass. And this is the same idea behind counterdiabatic driving. It's basically a way to move a system immediately to an end state by going through a different set of states than you previously would have that will approximate the set of states that it would have gone through before. Okay, so like I said, this is kind of mind-bendy. I feel like I didn't say that clearly. But I think if I go on to the data example, it might make more sense. So yeah, let me talk about how we actually tested this out. And I will try explaining counterdiabatic driving again when we get to that part. Okay, so if we want to do this in practice, I said that these different landscapes might, for instance, be drugs. And so we tested this out using data from a malaria resistance fitness landscape from Brandon Agbonu. And so what this is as before, we've got a set of 4 genes that can each be present or absent. Previously, we had antibiotic resistance genes. These are malaria drug resistant. These are genes that confer resistance to drugs that treat malaria. These are genes that the malaria might have. We don't want it to evolve resistance. And so this is an example of how that fitness landscape is connected. And then these curves here are showing fitness on the y-axis and concentration of this malaria drug, pyrimethamine, on the x-axis. And so this is actually a super incredible set of data, because if you think about it, each position along this x-axis is basically its own fitness landscape. And the cool thing about it is that the different genotypes shift in which one is better based on the concentration of the drug. And like they shift a lot. So seeing this paper the first time was kind of mind-bending to me, because I'd been thinking about each drug as a fitness landscape, but really this is saying each concentration of each drug is a fitness landscape. So that's a cool side note. But so this is the data set that we used. And what we wanted to know is if we're trying to apply different concentrations of this drug in order to steer this population through this fitness landscape, is there a way that we can do it faster than infinite time? And so here we have a set of drug dosages applied over time. Again, this is assuming we just apply whatever drug dosage we want at whatever time we want, which in reality isn't true, but we're not going to worry about it for now. And so this is representing the sequence of fitness landscapes that we would apply if we had infinite time. So actually applying this drug on this time scale is not necessarily going to work, because there is that lag between when we introduce a new environment and when the population reaches equilibrium for that environment. But this is the sequence we're going for. So what counterdiabatic driving allows us to do is derive a different, so this is just an example of a population evolving according to that drug ramp. So momentarily we're going to start increasing the concentration of the drug. So you'll see that it starts basically evolving more resistance. It shifts to these different alleles, genotypes. But you can see as we start increasing the concentration more, we end up with this lag. So sorry, I should have explained this better. The blue circles here represent the observed population distribution, and the red is what we expect at equilibrium. And you can see there was that really big lag at the beginning between the equilibrium and the observed, which implies that basically we hadn't reached the equilibrium yet, and it wouldn't have been safe to apply the next drug. And so whenever you see that lag, that means we're introducing more noise than is ideal if we want to be able to apply this in a precise way. And so our goal is to eliminate that lag. And so what counterdiabatic driving allows us to do is basically derive a different sequence of drug doses to apply. And what this sequence of drug doses is going to do is it's actually going to cause the population to immediately reach what would have been the equilibrium for the original drug dose that we were administering at that time. So it's kind of like tilting the tray. We're applying a force that is different than the one we would have applied in order to make us be able to safely go through that same equilibrium point without introducing noise. Just gonna pause there and let that absorb, because I know this is a little trippy when you first see it. And so just to prove that that works, so now we've got a comparison between the control, where we're not doing counterdiabatic driving, and an analogous counterdiabatic driving prescription. And you can see that when we do the counterdiabatic driving, we don't have that lag. We are always at what would have been the equilibrium for the initial drug sequence that we started with. And you can see the control just takes a really long time to catch up. So yeah, the conclusion here is that this works. Counterdiabatic driving can give us a really fine level of control of the rate at which the population is evolving through these fitness landscapes in response to our pushing it. And so this preliminarily seems like it solves this problem. So we haven't done this in the wet lab yet. So far, this has only been computational, but there was an effort to be trying it in the wet lab, and then we had to shut it down because of the pandemic, but I believe it's starting back up. So hopefully we'll have some wet lab evidence that this works soon as well. But the computational results were really encouraging. It's not that often that I get some data off the cluster and I look at it and it's just immediately perfect. But for this, it was. Like, it just worked on the first try. We didn't have to fiddle with it at all. So that gives me a lot of confidence in this technique. Okay, so that is the first story I wanted to talk about. I'm just going to pause for a second to give people a chance to ask any questions that they might have about this part. And obviously, there'll be more time for questions at the end. But if anything is confusing you.
Yeah.
Yeah, Simon?
Yeah.
So could you talk a bit about the population dynamics that go on during the control and the counterdiabatic? Because I would have thought the risk would be the population would crash or even go extinct if you're applying this huge spike of drug concentration. I don't know if that's a correct assumption, but did you observe any interesting population dynamics?
That's a great question. So the funny thing, right, so in my research, I'm constantly switching between situations where it's bad to kill the population and situations where it's good to kill the population. We were kind of doing this work from the perspective of preventing drug resistance. So killing the population from this mindset wouldn't have actually been that bad of a thing.
Okay.
We also never did, though, because we were just dealing with a parameter range where the population was unlikely to die because it was too resistant to the drug for it to be completely wiped out. But you're absolutely right that in different parameter ranges, you could kill the population. And if it's a population that you don't want to kill, that really just highlights even more the importance of having a fine level of control here. So the math to derive the counterdiabatic driving solution does take the population size into account, and it basically models the population size that you'll get in response to these perturbations.
Yeah.
And so we didn't try to do it here, but I think from that you should fairly easily be able to like determine what the risk of killing the population is.
Right.
And so you could be careful to steer away from that if it were important.
Yeah. So are you effectively deciding when to put bottlenecks on the dynamics using a mathematical model?
So I wouldn't say that it's so much the bottlenecks as just you're deciding when to change which genotypes are fitter or less fit. It's true that you're putting bottlenecks on it because, whenever you make the current most popular genotype less fit, you're going to kill a lot of things off.
Yeah. Yeah.
But I tested this computationally with and without bottlenecking. It definitely did affect the dynamics a little bit. I have a graph somewhere. I'm not sure if I could pull it up, but it didn't like have a profound effect. Like it didn't really make it into the paper because it didn't change things that much.
Right. Yeah. It just helps me understand because it's complicated, isn't it?
Oh, it's very complicated. Yeah. Yeah, that's a great question. Cool. I see we have another question in chat. Pardon my naivete, but am I approximately correct to say that a CD method is too analytically determine the optimal constraints-led approach to system performance? That's a good question. Okay, wait, let me think through that. Analytically determine the optimal constraints. Okay, I think partially. So I think you're right that that's ultimately like what we would be determining if we just had a set of fitness landscapes and we were trying to go from that to a fast sequence of fitness landscapes to apply in order to get the population from a start point to an end point. I think it's really combining this step with the step before it, because the step before it that I didn't talk about is the step where we figure out what the sequence of fitness landscapes that we need is. And this step is just trying to speed it up. And so I think finding kind of the optimal constraints is closer to what was happening in the step before. But I guess that there's also, it's not totally wrong to say that here. Yeah, okay. Now that I think about it more, yeah, I think that is a correct thing to say. Cool. Okay. And question from Alistair. This is very interesting. Thanks. Does CD only apply when there's a more extreme version of a target environment, such as an increased drug concentration? Or have you thought of ways it could apply more generally, i.e. when there is no more extreme version? Great question. Yeah, so this is something I've thought about a lot. We haven't tested it yet. Yeah, I think the way that we think about fitness landscapes and like sequencing them evolved a lot during this project. And so, yeah, when we started it, we definitely were thinking about drug concentrations as something where you can make it more extreme or less extreme. But as we were doing this, we realized that that's very much a simplification in that, the ordinal fitness ranking of these different genotypes changed as we changed the drug concentration. And so really each of these concentrations is its own fitness landscape. And so, yeah, so doing it with different drugs, with the same drug at different concentrations is definitely like the neatest way to do it mathematically, because even though it's different fitness landscapes, it's fitness landscapes that are like kind of related to each other by a continuous variable, which is just helpful for the math behind this. That said, so the data set that we used to get this fitness landscape from with the malaria drug resistance actually had 2 drugs in it. So I talked about pyrimethamine, but there's another one called cycloguanol. That doesn't really matter. It's just a different set of fitness landscapes related to drug dose. And we've done some preliminary work with actually swapping between landscapes from the 2 drugs. It's still messy because we're not sure what to think about the combined effect of the different drugs. But just in terms of being able to swap between completely different landscapes that aren't more extreme versions of each other, we have been able to use the combination of the 2 drugs as a way to kind of more precisely steer. So long answer, but basically, yes, this should work when it's not just a more extreme version, but we haven't tested it a ton yet.
Can I ask just as a follow-up to that, because I don't think I fully got it. Does that mean that sometimes the CD algorithm will go down as well as up in terms of the concentration in order to get to that point faster?
It could, yeah.
Okay.
Yeah, I don't think I've seen a situation yet where it's done that, but it's absolutely possible.
Thank you.
Yeah, no problem. Awesome. Yeah, thank you for the questions. Are there any more? Cool. Okay. So in that case, let's move on. So that was the nice, neat, fine control, well done math, good theory, elegant top-down solution. Now let's talk about the opposite. So yeah, a lot of the time I think of my work as trying to bring kind of nice, elegant theory together with messy reality of these just super complex dynamics of these systems. And so I'm kind of attacking it from both angles. So now I'm going to talk about the other angle. So this is work that it's going to seem like this is work that I started after the work I just talked about, but I actually started before and I've been working on it the whole time, and it's just much harder and takes much longer because I'm still working on it, and it is slow going. But I think it's also really important for us to understand. Okay, so in the previous talk or portion of this talk, I was talking about a situation where we were exogenously controlling the fitness landscape. You know, we were externally imposing different fitness landscapes on the population, and we weren't worrying about the fact that there might also be changes in the fitness landscape based on the population. Now I'm going to talk about the much more common situation where the fitness landscape is changing itself because there's an ecological community. So yeah, I'm going to talk about how we can predict evolutionary outcomes in these ecological communities. And specifically, the question I'm interested in is, how will the structure of an ecological community influence evolution in that community? And this is really important to understand to, again, solve this overall overarching question of how do we steer evolution? Because another point of control that we have might be setting up the structure of the ecological community. And maybe for these complex endogenously changing fitness landscapes, that's the best that we can do, because we can't really control the fitness landscape like I was talking about for the previous type of control. Okay, so this is a super open question. And the first challenge in answering it is just finding a good set of qualitatively different ecological communities to study. And so I'm briefly going to talk about the system that these ecological communities come from. So basically, I was able to find a good set of ecological communities to study in the context of evolutionary computation. And so at the beginning of the talk, I mentioned that evolutionary computation is a situation where we are using evolution as a machine learning algorithm. This is an example of an antenna that was evolved using evolutionary computation, just to kind of show that it works. So it's for NASA. And so yeah, evolutionary computation basically creates a bunch of populations and evolves them. And so in addition to using it as a machine learning algorithm, it can be a really great model system for studying eco-evolutionary dynamics. Then just a couple of things that you need to know about evolutionary computation for this to make sense is that the primary way that evolutionary computation goes wrong is when it runs out of diversity. Basically, if the whole population ends up having the same genotype, for instance, then it can't really evolve anywhere. So as a result, there are a truly ludicrous number of techniques for maintaining diversity in evolutionary computation. This is a small subsample of them. The point here is just that there are a lot, like just a ludicrous number. And the other key that I want you to just know, so that what I'm about to say makes sense, is that basically all of these techniques create ecology. I'm not going to belabor that point because the point of this talk is not to convince you that these techniques create ecology. Feel free to ask me about that later. I would love to talk about it. But just as an example, on the left here, we have some math that was derived to predict the circumstances under which two genotypes might coexist in fitness sharing. Fitness sharing is just one example of one of these algorithms that maintains diversity in evolutionary computation. And on the right, we have some math that was derived at actually around the same time to predict the situations under which two species could coexist in ecology. And the point I want to make here is just that this math is the same. And these authors did not originally write it the same. I did some math in this preprint that is still not out, where I showed it's the same. Feel free to ask me for the link if you want to read about it. But the point I want to make here is just that when I say that there is ecology happening in these evolutionary algorithms, I'm not exaggerating. There's literally mathematically the same ecology happening in them that is happening out in nature. So this is totally a legitimate model system to be using to study these questions. And it's also a good model system to be using because it evolves so much faster and it is so much more possible for us to measure the ecology than it would be in nature. Okay, so now that I have set the stage for what system I'm using to address this question, the specific question that I'm trying to answer right now is, do these different evolutionary algorithms with their different diversity maintenance strategies create meaningfully different ecology? And you don't need to worry about the fact that these are diversity maintenance strategies. You can just think about them as different ways of constructing an ecological community. So one way that we might try to understand the ecological community that's created in different contexts so that we can compare them and see if they're qualitatively different is with this concept called interaction networks. And so these basically are just networks that indicate how different members of a community interact with each other. So for instance, here, member A is harming member B, and that's indicated by this red arrow. And the fact that it's a large red arrow means that A is harming B a lot. And specifically what I mean by harming here is that B's fitness is lower when A is present than when A is absent. In this instance, B is also harming A, but much less. The other kind of interaction that can happen in these networks is a beneficial interaction, shown in blue. So here, B is helping C, which means C's fitness is higher when B is present than when B is absent. And so this is just a tool that we can borrow from ecology in order to describe what these ecological communities look like. So let's try it. And so yeah, the answer to the question of are these going to be different? Spoilers, is going to turn out to be yes. So here I've got just an example randomly generated population. And I'm going to try building the ecological community that would be present in that population under these 4 different regimes. So the first one is called tournament. It's kind of a control here because it doesn't actually create interactions. And so you can see basically there's not variation in this network. Everything harms everything a little bit because they all compete with each other. That is in stark contrast to oh, well, okay, not stark, but it's in a bit of contrast to fitness sharing. So fitness sharing is an example of a diversity maintenance algorithm that exists in evolutionary computation. You can see that once we change the way that we're calculating the fitness to implement fitness sharing, as opposed to the control tournament selection, suddenly there are some beneficial interactions. And also there's variation in the strength of the negative interactions. So just by changing the way that we're choosing who reproduces here, we're changing the structure of the community. And this gets even more drastic as we try some other evolutionary algorithms. So EcoEA is again just an example of a different evolutionary algorithm, but it produces a radically different community structure that's got a bunch of beneficial interactions and some much more intense negative interactions as well. And afterwards, if you guys want to talk about specifically what these different evolutionary algorithms are doing, I'm happy to. It's really cool, but it's not the point here, so I don't want to get too sidetracked. So that's clearly a very different community. And then another evolutionary algorithm that produces even more different communities is called lexicase selection. You can see this is characterized by having a very small number of very intense negative interactions. And so this is looking pretty compelling in terms of these different evolutionary algorithms creating communities that are different in meaningful ways. And indeed, if I try just generating 100 populations and applying each of those different selection regimes to each of them, I can see that these differences are consistent. So here I just counted up the total number of beneficial and harmful interactions in the networks for each of these evolutionary algorithms. And there are other ways that I could quantify them too, but this was just a nice straightforward way. And you can see that like the differences that I showed in that previous slide were very representative. They hold up across these 100 replicates. So these are ways of constructing communities that are qualitatively different. So, okay, great. I have now established that this is a viable model system. It lets us create the different ecological communities that we want to start from. And then the question that I said that we want to answer in the first place is, what are the implications of that for evolution? Is there a way that we can use this information to control evolutionary outcomes, or at least to predict them? So I'm still working on this. But the idea of how we might be able to measure this is based on phylogenetic metrics. So whenever we have an evolving population, we can represent it as a phylogenetic tree. So this is basically just an ancestry tree. So, you know, for instance, here in the center, we've got whatever the first member of the population was, and then we've got its two offspring, or sorry, I guess it would actually, no, sorry, it's 4 offspring, here, here, and here, and here. And then each of those offspring have offspring, and so on and so forth. So we can just draw this as a tree. And that's a very information-rich way of conceptualizing the population, but it's a little hard to compare statistically in a useful way. So we can extract summary measurements from these trees. There are a lot of them. The one I just want to call your attention to right now is richness, because that's the one that I'm going to use here. And that's basically just measuring the total amount of evolutionary history represented in the tree. It's just a nice way of kind of, yeah, getting at how much the tree has diverged. So based on that, my goal is to use that as a proxy for seeing if there are qualitatively different evolutionary dynamics in these different ecological communities. And so since this is an evolutionary computation system, I tried these in 3 different fitness landscapes from evolutionary computation. And so in the previous section, all the fitness landscapes I were talking about were drugs. Here, all the fitness landscapes I'm talking about are computational problems that you might want to solve. So the first one, squares, here is just squaring a number. So literally, we're just trying to evolve code that will take a number in and output its square. The collets is a more challenging computational problem where you're trying to like figure out when a sequence will converge. And then DAO is a practical real-world data interpretation problem. So you don't need to worry about the specifics of them. Just know that these are 3 different fitness landscapes. And for each of them, I tried letting a population under each of these regimes evolve, and I measured the phylogenetic diversity. And so that is the total amount of evolutionary history represented within the population, basically. And this is what I found. So there's definitely some variance by problem, but pretty consistently, we see that one of these selection schemes, the one called lexicase, so that was the one that created the small number of intense negative interactions, maintains a lot more phylogenetic diversity than the others. And then seems like maybe EcoEA maintains the next most. That was the one that had kind of the mix of beneficial and positive interactions. But that seems like it varies more by problem. And so I'm still trying to think through all the implications of this, but it's pretty solid preliminary evidence that there are evolutionary consequences to these different structures. And what I'm working on now is kind of zooming into a smaller temporal resolution to figure out the mechanics of why exactly this is happening. But I don't have answers for you yet. Maybe in a few weeks, we'll see. So in conclusion, I think this is a really promising system to use to address this very messy question. And it does look like there's something here. These ecological communities are leading to different outcomes, and I'm excited to continue this work. So with that, I just want to acknowledge a few people. So first off, all my co-authors on the Counterdiabatic driving project that I talked about first. Like I said, this was a huge interdisciplinary collaboration. So all these people contributed a lot to this project. I'd also like to particularly thank Jake Scott, my postdoc advisor, and Charles Zofria, my PhD advisor. Since I'm still in my first year of my faculty position, their thinking and mentorship has been incredibly informative in coming up with all of these ideas. And then also just like to thank a variety of funding sources and communities I have been a part of, all of which have helped me to start thinking about this problem. And thank you all for having me and for listening.
Thank you very much, Emily. That was absolutely fascinating. I'm going to switch the recording off now, so that the rest of the questions won't get recorded, but thank you very much.